Rivus Pharmaceuticals, and I believe they are ready to roll.
Great. Good afternoon. My name is Alan Cunningham. I'm Rivas's President and CEO. Appreciate the opportunity to participate in the showcase and tell you about our company and plans. I'll share some slides here. So Rivas is focused on advancing a new class of medicines called controlled metabolic accelerators, or CMAs. They're designed to improve metabolic health and reduce cardiovascular risk and mortality. Our lead program, 826, it's an oral small molecule. We've completed phase 1 single ascending and multiple ascending dose trials, and more recently a phase 2A metabolic study. And I'll review some of this data. But the data indicate that 826 has the potential to be a leading single agent in cardiometabolic disease via its fat-selective weight loss and improvement in key markers of glycemic control and inflammation. So we're seeking to address this common problem in cardiometabolic disease, which is energy excess, this imbalance of energy intake and energy expenditure, which leads to common conditions of dysmetabolism, high blood glucose, high triglycerides, high blood pressure, etc. There are basically two ways to address this energy excess. You can either reduce the input or you can increase the energy expenditure. And we're focused on the latter, and there are key advantages of doing so. And it starts with fat-selective weight loss while preserving skeletal muscle mass. And this is in contrast to current approaches that, such as the GLP-1s and their combinations, that induce caloric deficit via appetite suppression. And these approaches result in a reduction of muscle mass, which is significant. Up to 30% of the total weight loss of GLP-1s can be skeletal muscle mass. And importantly, the weight regain after caloric deficits is almost entirely fat. So once you lose that muscle mass, it's hard to get it back. And as the prior speaker noted, particularly in elderly sarcopenic populations, that preservation of muscle mass is quite important. So by targeting energy expenditure, we can also increase resting metabolic rate. We can improve metabolic flexibility, as well as compliance and sustainability, since caloric deficits are difficult to maintain over time. So our CMAs leverage a mitochondrial uncoupling mechanism. Uncoupling, or proton leak, it's a natural mechanism by which the body generates heat. And 30% of the calories that we consume go toward uncoupling. Uncouplers increase proton leak across the mitochondrial intermembrane space, bypassing ATP synthase. And because these protons are leaked, mitochondria oxidize additional carbons to maintain the same amount of protons for ATP synthesis. The result is increased fat oxidation while preserving muscle mass and ATP. Turning to our 2A metabolic study and some highlights, this was a double-blind, placebo-controlled study in high BMI subjects with evidence of liver fat greater than 8%, which determined by MRI-PDFF. Patients were randomly assigned 826 at 150, 300, and 450 milligrams versus matched placebo, and they're treated over an 8-week period. We also had subjects with elevated HbA1c included in the study, including pre-diabetics and diabetics. And these subjects were distributed equally across these groups, approximately 40% per group. And I'll note some compelling differences that we saw in this particular patient population. So the primary endpoint of the study was a reduction in liver fat, which we saw across all doses of 30 to 40%, whether normal HbA1c or elevated HbA1c. And this reduction in liver fat saw high responder rates of 70% or more at the mid and high doses. And the responder rate is it's per the Lumba et al. Criteria, defined as having greater than 30% relative reduction in liver fat by MRI-PDFF. So turning to weight loss in this study, in the 8-week period, patients at the high dose lost 6 pounds of body weight, which was almost entirely accounted for in fat. And intriguingly, this fat loss was primarily biased toward visceral fat. This is important because visceral fat plays an important role in cardiometabolic disease. So this combination of being fat selective while being more highly targeted to visceral fat is really unique to this approach. So in the patients that had elevated HbA1c, we actually saw amplified weight loss. A placebo-corrected 10 pounds of weight loss at 8 weeks and 11 pounds of fat loss. And it's worth mentioning here that 826 had an even greater impact on visceral fat in these subjects with elevated HbA1c. So via this fat oxidation mechanism, 826 burns fat in the absence of diet or exercise. And I may have failed to note that for this study, we were very careful to note that that patients should avoid any behavioral changes. So we advise them not to change if they're on a diet or they were exercising, keep doing what you're doing. If they weren't, keep doing what you're doing. So we also saw improvements in glycemic control as measured by reductions in glycated albumin in this 8-week period, as well as reductions in systemic inflammation as measured by high sensitivity C-reactive protein. In this measure, 826 had a placebo-adjusted 60% reduction in this important inflammatory marker without being an anti-inflammatory. So in our clinical trials to date with 826, there have been no serious adverse events. 6 subjects have discontinued early, 3 on treatment, 3 with placebo. There have been no discontinuations at the high dose. AEs were predominantly diarrhea and flushing, most often mild, which occurred in 35% and 25% of the 826 subjects respectively. There was evidence of reduction in diarrhea over time. There was no increase in body temperature, heart rate, or respiratory rate, and 826 was well tolerated. So building on this phase 2A data, we've now lined up the next phase of development for 826 and our company. We have just initiated a 2A study in heart failure with preserved ejection fraction, or HFpEF, and we have a plan 2B obesity study where we'll file an IND early next year. Additionally, we have a robust discovery program for the development of additional controlled metabolic accelerators, and we're looking to bring another candidate to the clinic in the next 12 to 18 months. We're very keen on HFpEF. These patients are obese. They have hypertension. Half of them are diabetic. Our data indicates that we can treat all 3 of these pathophysiologies, but also reduce visceral and epicardial fat, reduce ROS and inflammation, and ultimately we're hoping that sustained weight loss can improve quality of life in these patients and exercise tolerance. Of course, for obesity, there's an exciting opportunity given this differentiated mechanism for increasing energy expenditure. And our 2B study will treat for 12 months. We'll have the primary readouts will be weight loss and reduction in A1C. And this is exciting. We know that sustained weight loss can lead to remission of diabetes, and we think that we can bring these patients to a place of improved metabolic flexibility. So in summary, for HU6, it burns fat in the absence of diet or exercise, it preserves skeletal muscle mass, it has amplified weight and fat loss in patients with elevated HbA1c, which is unusual. The GLP-1s, for example, have approximately half of the weight loss in patients with elevated HbA1c. HU6 has a significant impact on visceral fat, and it reduces glycated albumin and markers of inflammation, and it's a once daily oral that's well tolerated. So happy to answer questions.
Thank you so much, Alan. First of all, congratulations on the significant round of funding that was just closed. So I don't know
Yes,
if you had a little party at the office for that, but hopefully so.
Yeah, we're very excited. We have a terrific group of investors who share our vision for treating cardiometabolic disease, and we're well funded to execute on the next phase of clinical plans that I've just outlined.
Absolutely, and to your point, your investors, I believe the round was led by RA Capital.
That's correct. RA Capital is our lead investor, and Bain Capital Life Sciences and BB Biotech also participated. Our existing investors, Longitude Capital, Medici, and RX Capital. So really great to have support, such strong investor support as we pursue these large indications.
Yeah, and this is absolutely a very sophisticated group of investors. And that leads me to ask the question in the chat, sort of the elephant in the room, which is, I believe HU6 is a DNP derivative. And as such, the question is, how has Rivus overcome the safety challenges that agent historically had?
Yeah, so HU6 it's a new chemical entity. It does metabolize to the active DNP. Of course, we have built in key design features to address the toxicities of traditional uncouplers, and yet we've maintained the compelling efficacy for this fat oxidation. So basically, there are two key ways in which we've designed this compound to be safe, and that is we've reduced the CMAs, so it has a nice flat PK curve, and it also saturates at higher doses, so it has a cap on the exposure of the uncoupling. So we have this nice, steady, mild uncoupling that can benefit these patients.
Fantastic, fantastic. That's great news, and clearly that approach has been validated by that large round of funding. And I'm curious, and I suspect the audience is curious as well, what was the data or the moment where the investors in the syndicate said, yep, we're in? What do you think was that trigger?
I'd like to say there was one moment. Of course, these, as you noted, these are sophisticated groups, and we have a very complete program with a full, all of the preclinical and non-clinical data leading to the IND, the data from our long-term tox studies in multiple species, the single ascending and multiple ascending dose data, and then the 2A data, all of which was reviewed over a period of time and very closely. So there's no one particular moment. It's really the totality of the program and its potential and the differentiated approach, again, that we can have this fat selective weight loss while preserving skeletal muscle mass, and our approach is fundamentally different from current approaches in the obesity space, and I think the investors recognize that opportunity.
Yeah. Well, fantastic. Congratulations again, Alan, and we'll look forward to the launch of your next trial.
Great. Thank you so much. Again, appreciate the opportunity to participate today.